Sensory modalities then. What can we sense? These are the main conscious somatic sensory modalities. The dorsal funiculus we met yesterday, dealing with touch, pressure, joint proprioception, and the spinothalamic tract, dealing with pinprick pain, heat and cold. The spinocerebellar tract, dealing with muscle proprioception, I've put in grey. It is a sensory modality, but it's not something that reaches conscious perception. It's not easy, and for many people not possible, to think about what kind of tone you've got in your different muscles. In the next couple of lectures, although we're not going to deal with it today, we're going to be dealing with the modality of pain. And for completeness for this diagram, I've put on the special senses that we've already dealt with last semester. So those are the things that you can sense. That is the information that you use to build a picture of your environment. That's how you know what's going on in the world. In some books you will see the term kinesthesia. It's not a term that I use very much, but it is out there in the big wide world. It literally means movement sense. And it's a kind of conglomeration of muscle and joint proprioception. Let's start at the start and have a look at some of these sensory modalities and start with the receptors. Receptors are interesting things in that they all have a nerve ending which converts something to a rate of generation of action potentials. And so for nociceptors, those tend to be bare nerve endings in tissue, right up to the big Pacinian corpuscles that look like onions and have a nerve ending embedded in a more complex structure. And the structures around them modulate the kind of thing that these receptors respond to and modulate the way in which they respond to it. And the main modulation is in the speed of adaptation. So if we classify the receptors, we can start off with the simplest, the free nerve endings, which are slowly adapting. In the middle, we've got the medium complexity neurite receptor complexes like the Merkel cells, which are slowly adapting, and right at the top end of the complexity scale, we've got the complex laminated receptors, and they are rapidly adapting. So they will respond to change in the environment more than magnitude of stimulus in the environment. The free nerve ending is embedded deep in the middle and runs through it. And what happens is, as a force alters the shape of the skin that the Pacinian corpuscle is embedded in, it alters the shape of this outer layer, and that tends to alter the shape of all the inner layers. Altering the shape of or distorting the free nerve ending is what generates the action potentials. But because this is such a big spongy affair, once the distortion's taken place, the inside fairly easily moves back to its original shape, and so stops distorting the nerve fibre and it stops responding, hence the adaptation. Another example of adaptation is here, and this is a section through a whisker sinus of a rat. Here's the whisker sitting in the middle in its follicle. There is a blood-filled cavity or sinus that runs around it, and in this membrane that goes around the edge there are free nerve endings. And the free nerve endings in this model are responding to the movement of fluid in that whisker, so that when the whisker touches something as the rat's exploring its environment, it moves the whisker in its sinus. That causes disturbance in the fluid-filled area here, which causes disturbance in the membrane and distortion of the free nerve endings, causing action potential generation. And so there we've got a very acutely sensitive and delicate sensory mechanism. An adequate stimulus is the classification of the stimulus that preferentially affects that receptor. So that, for example, the adequate stimulus for a chemoreceptor is a specific chemical. And the function of that chemoreceptor will depend on both where it is and the particular chemical that it responds to. So when you're thinking about the adequate stimulus, you're thinking about the thing that does it for the receptor, and when you're thinking about the function, that's a combination of what the receptor's looking for and where the receptor's been placed to look for that. You will remember, I hope, from your very basic neurophysiology and also from some of the special sense stuff that we did last semester, that when we're talking about action potential generation, we're talking about changing the resting membrane potential to threshold to generate an action potential, and that the further beyond threshold the resting membrane potential goes, the shorter the refractory period between action potentials. So that what we've got here is an analog change, which can be delivered by all sorts of physiological mechanisms, which is leading to a frequency change in the action potentials travelling up the sensory nerve. And so it's the frequency of action potential generation that's important for the transmission of a sensory stimulus, a sensory modality. And that brings me to lateral inhibition, which we talked about under the guise of vision last semester, and which I want to go through again with you under the guise of touch this time, hence the Pacinian corpuscles, to talk about how lateral inhibition in the various spatial senses that use lateral inhibition reduces the amount of information that needs to travel up to the central nervous system, and at the same time enhances the sensitivity of the sensory system to the environment. You'll remember last time we talked about the concept of background rates of action potential generation, and that a resting sensory nerve is not really resting in the sense that it's not doing anything, but it's resting in the sense that it is generating a certain rate of action potentials. And that rate of action potentials can increase if that nerve is facilitated, or decrease if the nerve is inhibited. We looked at the effect of stimulating the central sensory field in a given area, and we said that the effect of stimulating that central field was that there was an increase in the rate of action potential generation in that field that was stimulated, and a decrease in the rate of action potential generation in the surrounding fields. And that's the basis of lateral inhibition. That's what we're talking about. We're talking about a sensory stimulus inhibiting areas that are lateral to it. And whether we're talking about that in the visual field or in the somatosensory field, then that's the mechanism that we're talking about. We're talking about stimulation at one area inhibiting lateral areas. Why is that a good thing? Well, we went on to consider the effect of a stimulus over a whole area of these receptive fields, and to look at the effect that that would have. And you can see here that if you stimulate this area, the response that you get in terms of action potential generation is an increase in the areas within the zone of stimulation but adjacent to its edge, a decrease in the areas outside of the area of stimulation but adjacent to its edge, and no net change over the rest of the areas, either because these areas are both stimulated and inhibited, or because these areas are not receiving any stimulation or inhibition because they're away from the area of stimulus. And if you imagine that diagram getting larger and larger and larger, you can see that because the perimeter increases in a linear sense and the area increases in a square sense, the bigger the area of stimulation, the more savings you get coding for that area of stimulation in terms of number of receptive fields that are conveying changed information up to the central nervous system. So this is a system by which we can get more efficiency out of our sensory systems. We send less information up to the central nervous system, but the central nervous system interprets that information to get the same amount of knowledge of the environment. There is another advantage of lateral inhibition. Here is a diagram of, if you will, the neural space of a piece of skin, i.e. the part of the sensory central nervous system that codes for sensation in a particular piece of skin. And so here in a two-dimensional representation of that space, i.e. a square area of skin, we've got neural activity, change in rate of action potential generation, resulting from a single point stimulus. And you can see the typical pattern represented in a slightly different way. Here at the centre where the stimulus is, we've got facilitation and an increased rate of action potential generation. Around the periphery, we've got a decrease in the rate of action potential generation, and away from the stimulus we've got no change in the rate of action potential generation. Now that becomes interesting when we look at two separate point stimuli, slightly separated in space. So in this diagram we've got the same neural space. This time a two-dimensional slice through it has been taken just to make life easier so that we can see what's going on. But if you want to, you can mentally project that into a sort of three-dimensional pattern. And at the top here, we've got the resulting stimulus that you would get from two separate point stimuli, point sources, if there was no lateral inhibition. And you can see that you can detect the two separate stimuli, but there's not a lot of contrast in the area between them. And so there's not a lot of neural differentiation between those two separate points. If we add lateral inhibition into this, you can see now that at the edges we've got this reduction in action potential generation that we didn't have in the other model. But also look at this great trough that we've got between those 2 points. We've got much better spatial resolution for two separate stimuli. And so when we utilize lateral inhibition, we're better able to discriminate two separate senses in space as they get closer and closer together. And the point at which they merge into one stimulus becomes a lot closer. So those are the two advantages of lateral inhibition. Less information goes up to the central nervous system, but the central nervous system gets more value from that less information and gives you a sensory system that is in effect more subtle and more able to get subtle changes in its environment. What I want to talk about for the rest of this lecture is to talk about the sensory tracts that carry that information up to the higher centres of the central nervous system. And because today we're dealing with sensation, I'm going to be concentrating on the dorsal funiculus and the spinothalamic tract, which together form the major sensory system in the central nervous system, which is known as the medial lemniscal system. Let's have a look at the dorsal funiculus, which you'll remember is coding for touch, for pressure, and for joint proprioception. And here it is in the spinal cord. Caudal spinal cord at the top, going through to cranial spinal cord at the bottom. Notice the number of fascicles within the dorsal funiculus increases as we move cranially through the central nervous system. Medially here, we're coding for caudal areas, and laterally, we're coding for cranial areas. And you can see that as these fibres come into the dorsal horn, a branch of the nerve fibre shoots off medially, joins the dorsal funiculus, and carries on up the central nervous system all the way to the medulla. And that as subsequent nerve fibres join this pathway, they join it in an organised way, and so we get this movement, this somatotopic localization with caudomedial craniolateral organisation. Here it is larger and in glorious technicolor, and so you can see that the gracile fascicle is carrying information from the sacral and lumbar sections of the spinal cord, and the cuneate fascicle is carrying information from the thoracic and the cervical segments of the spinal cord. That means that if you look at the caudal segments of the spinal cord, you only have the gracile fascicle. The cuneate fascicle doesn't exist until you get up to the thoracic areas, because everything is joining in a logical manner. The spinothalamic tract is coding for pinprick pain, heat, and cold, and is similar to the dorsal funiculus in that information is going up to the higher centres of the central nervous system, but different in various ways. Firstly, it's different in that the ascending fibres are not the primary sensory neuronal fibres, but there is a synapse between them, and so they are effectively interneurons, secondary sensory fibres. Secondly, the fibres join the tract not from the lateral, travelling medially, but the reverse, from the medial travelling laterally. And so we've got this same organised addition to the spinothalamic tract, but this time we're coding laterally for caudal stuff and medially for cranial stuff. Because the fibres come from medial to lateral to join the tract, and they line up logically, it follows that they have to code in the opposite direction. The last thing to say is that the spinothalamic tract is a largely, though in our species not an exclusively, contralateral system. So that as the dorsal funiculus is ipsilateral, on the same side as the stimulus, the spinothalamic tract is largely contralateral. The textbooks that you read, most of them will tell you that it's exclusively contralateral, and in man and probably the primates it is. But in our domestic species, there is a fairly significant, although not as large, ipsilateral contribution as well. So that in the domestic mammals, it's a bilateral system. Again, let's look at that somatotopic localization in more detail. And you can see that we've got the same concept that in the cervical spinal cord, we can draw a picture of the animal onto this tract, but this time the caudal areas are lateral and the cranial areas are medial. And you don't have to remember that, you just have to remember that the fibres are added in a logical way, and so when the fibres are added from the medial side, you get this coding, and when the fibres are added from the lateral side, from the outside, you get the opposite coding. That's going to cause us some interest in a couple of slides' time when we try and put these two systems together so that we get one logical system at the level of the cerebral sensory cortex, with one area of cortex dealing with one part of the body. So first of all, let's put some labels on this to look at the segments of the higher centres of the central nervous system. So here we've got the medulla oblongata, just at the top of the spinal cord. This is where you can expect to see those tracts, the spinothalamic tract and the dorsal funiculus, emerging into the brain. Here we've got, slightly higher up in the brainstem, the cerebellum and the pons. And here we've got a section through the midbrain, the thalamic nuclei, and the cerebral hemispheres wrapped over the top. Here we've got the cuneate nucleus and the gracile nucleus, which are nuclei, so there are cell bodies there, which sit at the top of the tracts of the dorsal funiculus. They're sitting at the top of the cuneate and the gracile fascicle. Here we've got the spinothalamic tract, coming up more or less in situ, in the localization where it went up the spinal cord. And here we've got the medial lemniscus. Now, the problems that we have are that we've got to get an ipsilateral dorsal funicular system lined up with a largely contralateral spinothalamic system into this medial lemniscus to ascend further on. Going further up, and again, just for localization, we've got the trigeminal nerve and the nucleus of the trigeminal nerve. Here, the medial lemniscal system, and those both sit under the fourth ventricle. And then at the level of the midbrain and the cerebral cortex, we've got here the primary somatosensory cortex, where all this information is going, the lateral ventricle, and, just for localization's sake, the lentiform nucleus. So let's look at these two systems each in turn and see how they get between the spinal cord and the sensory cortex. Here are the sensory neurons of the dorsal funiculus. Remember, those are still branches of the primary sensory neuron. They've come all the way from the tissues, so that if they happen to be in the hind foot of a horse, those fibres at this stage could well be 2, 2.5, even 3 meters long. They're going to be long fibres at this stage. They synapse in the gracile and cuneate nuclei and become secondary sensory fibres. At this point, the fibres decussate, they cross the midline, so they've become contralateral and come into the same orientation as the fibres in the spinothalamic tract. And as they do that, they also cross over each other so that their somatotopic localization lines up with the spinothalamic tract, so that these fibres are now in line with the spinothalamic tract. They travel up as secondary sensory fibres through the medial lemniscal system up to the thalamic nuclei, and there they have another synapse. Remember, in the first lecture I mentioned that very few fibres get into the cerebral cortex without synapsing in the thalamus. And so here, these neurons synapse into tertiary or third order sensory neurons, and they travel up into the cerebral cortex. Now notice that they appear to have again reversed, in the sense that a lateral fibre in the medial lemniscal system follows this path and becomes more medially located on the somatosensory cortex. And a medial fibre in the lemniscal system takes this path and ends up being more laterally oriented in the primary somatosensory cortex. And that, in a sense, is a bit of a cheat, because embryologically what appears to happen is these cortexes sprout from this area of the brain, but there's no room for them down here. And so in mammalian species where those cortexes are large, they fold out on themselves and fold round. So what we're seeing here is not a change of orientation within the neural tissue, but we're seeing that the neural tissue kind of folds round on itself and takes it with it. So in effect, we've got one decussation, one crossing of the midline, and one change in somatotopic localization to line up with the spinothalamic tract. Okay, let's go on to look at the spinothalamic tract. The spinothalamic tract is already, largely speaking, contralateral, and I haven't put the ipsilateral branches on here for simplicity's sake. These fibres travel right the way up to the thalamic nuclei, but remember that these are already secondary sensory fibres. These are not the original fibres that came from the tissue. They've already synapsed in the dorsal horn of the spinal cord. So once we get beyond this level, we've got an orientation in space. Both sets of fibres from the dorsal funiculus and the spinothalamic tract are lining up with each other in a spatial sense. And we've got an orientation in the sense that these are both the second neurons in the pathway. The neurons come down and have a synapse in the thalamus and do exactly the same round the corner trick to give you exactly the same lineup in the primary somatosensory cortex. So what we've got here is a system by which information from one part of the body takes various pathways up through the central nervous system, but everything comes out in the same place right at the top. And so we've got a body with a certain orientation at the bottom. We've got a primary somatosensory cortex with the same sort of localization at the top, and everything in between is a little bit different, a little bit the same between the two systems. So when we put the whole thing together, we get this diagram that at first look appears rather daunting, does to me anyway. But you can see that what we're doing here is we're bringing this sensory information together into the same pathway, altering its orientation so that everything lines up together. So we've got a pathway or two pathways which are travelling up different parts of the spinal cord, doing it through different routes but both logical if you look at the way that the fibres join the two tracts. And by a little bit of sleight of hand here in the medulla oblongata, aligning themselves so that they both end up in the same part of the somatosensory cortex. One thing I didn't mention earlier is the cranial nerve contribution, and you can see here the contribution from the trigeminal nerve, where both sets of fibres travel together because they don't have to go up the spinal cord, and they've already got the medial lemniscal system here which they join straight into. But for the spinal senses, the most important part of what I've been saying today, we've got these two systems that are lining up to become one system. So that when they get to the higher centres of the central nervous system, we can look at them and there is a certain aesthetic efficiency about the way that the different areas within the somatosensory cortex code for information from different parts of the body.